So I think before I introduce about the assay, I think it's important to address the elephant in the room, which is the why and when should one even consider performing such an innate immunogenicity assay. So it's actually coming down to the risk of the silent immunogenicity, where many of the biologics developer, generic peptide, okay, maybe not generic peptide, but biotherapeutic developers, they use animal studies to actually inform them about what are some of these immunogenic risks that the product carries. And more often than not, this animal data does not translate very well into the human data, as we most know. So then that leaves a gap, right, between the preclinical setting to the clinic setting. What is the innate immunogenic risk in the drug product? And is it worthwhile to risk all the time, money, and manpower committed and invested into this just to see an undesirable clinical outcome and then just to go back to the drawing board? Because that's a high-stakes situation. So the gap that we're referring here, it comes down to a number of these factors that could actually drive an innate immune response, such as your aggregation, formulation, host cell proteins, post-translation modifications, and the innate immune response modulating impurities, which for the rest of the presentation, I'll just term it as IIRMIs. So I really like what Dr. Danella has said, is that undetected risk is not the same as absence of risk. I paraphrase it, right, from what he has mentioned. So the whole point is to address how can we address this gap before we have the product reaches the clinic. So when the gap actually reaches the clinic, this is where the classic case study comes in, your TG-1412. There's a humanized anti-CD28 superagonist monoclonal antibody, right? When in the first in-human trial happened, there was a near-fatal cytokine storm in all 6 healthy volunteers within minutes of infusion. And this was clearly unexpected because they did not see any risk when they used an animal model to look at this. And then that begs the question, right? How can we then address it before it reaches the clinic to avoid such an unfortunate incident to happen again? So that's just one part of the drug modality. In that case, it was just a monoclonal antibody. But as we know, there are a bunch of different drug modalities out there. So what can then the risk possess that we need to be concerned about this innate immunogenicity risk? So these are some examples of a range of different drug modalities where such innate immune triggers can exist. So like your antibodies, it can be from an Fc-mediated activation or aggregation that can actually trigger cytokine responses, as we know. That's independent of the target engagement itself. And for your ADCs, right, if we ignore the antibody scaffold part, there's the linker and the payload components that you need to consider, whether it can actually be your innate immune triggers that can trigger the pattern recognition receptors, which we call it PR for short, and beyond the antibody backbone. So there are also delivery systems like in the vaccines itself, your LNPs, mRNAs, viral vectors, etc. Does the carrier itself actually carry such PR activating potential? This will also be an issue as well. And then you have your therapeutic peptides where, when it comes to generic peptides, we have like process-related impurities that could potentially trigger. Any of these impurities that trigger PRs would result in the unwanted innate immunogenicity risk. And then your novel scaffold proteins, you have your non-native structures, which is foreign, which can also be potentially immunogenic to the immune system. And lastly, you got the enzymes and replacement factors where you got species-specific glycan patterns and residual host cell proteins that can also introduce innate immunogenicity risk. So all in all, you may notice that the factors can vary, but the most important thing is how can we then assess these factors or risks as early as possible before it reaches the clinic so that we can do something about it before something not so great happens in the clinic. So now we can move on to the question of how can we assess then the innate immunogenicity risk at the preclinical stage, and that's where the assay actually comes in here. So introducing our ProStorm, which is actually a fresh whole blood cytokine release assay. So we basically recruit 10 to 30 healthy donors for each study, and then we will have laboratories to draw a 50 ml of blood. And then within 3 hours, this blood will be transported to our lab to actually incubate your test articles into the blood, and then we carry out a 24-hour incubation with the test materials. And afterwards, we will then harvest the plasma, quantify the cytokines based on the cytokine panel that we have, and the whole project will take about 5 to 6 weeks to complete, where we will actually have the report that's actually sent to you. And afterwards, what we like to do is to actually have a debrief call, because while the report is very detailed as it is, depending on your knowledge and understanding of it, the interpretation of results could differ. So the whole debrief is just to align everybody's understanding of the results. So the big question comes in, right? There are different ways that you can do it. You got PBMC-based assay, so why fresh whole blood? So the easy answer is, why not? Right? So in this case, the whole blood here, they have no bias that's introduced due to there's no cell purification or isolation. It's just undiluted fresh whole blood. There's no manipulation involved. And it also allows you to capture or detect neutrophil-mediated cytokine release, which is a signal that's absent from your PBMC-based assay. And the most important part is that it captures the natural ratios of all your different antigen-presenting cell subsets in the blood itself as compared to your PBMC. So that will allow you to, and in a way you can think of it that this is really the closest you can actually get before your drug actually reaches the clinic. That's in vitro. So this is a very simplistic summary of our assay workflow, which basically where we incubate the test article in the fresh whole blood. We do it in triplicate wells. After 24 hours incubation, we will actually harvest the plasma. We will pull the triplicates together for analysis, and then we run a triplicate analysis to actually have a 7 cytokine readout in this case. So you got IL-2, 4, 6, 8, 10, TNF, alpha, interferon, gamma. But of course, if it's required, depending on what cytokines you're looking for, we can always consider adding them in the panel. So going back to the risk of silent immunogenicity, the next part I wanted to highlight is actually more of the aggregation factor on how the extent of aggregation based on concentration can actually impact the innate immunogenicity. So this is a data that we actually done for market-approved antibodies where we have like Remicades, you got Herceptin, Vectibix that's actually included in this assay. And just to better understand, for everyone to understand, this is basically based on IL-6 readout. And here, every single dot here represents a donor response. And the red color bar that you see here, that represents the median cytokine response for the total number of donors that's here. So here we have the PBS, which is the phosphate bufosylin for the negative control, and we got staphylococcal enteroxin B as the SCB, that is the positive control. Now the interesting part is that you see the Remicade at 4 concentrations from 0.1 to 100 micrograms per ml, you see that the median response is actually near 0, so which is kind of expected. And then when you look at the aggregated Remicade, you notice something interesting happen, right? At 0.1 and 1 micrograms per ml, it still remains as near 0 response. But when it goes to 10 micrograms per ml, you see that the median response starts to creep up a little bit. And then when it comes to 100 micrograms per ml, the response is equivalent to the positive control. So this shows that the aggregation actually, it matters based on what is the concentration that you are using here. So, and non-aggregated antibodies here are near 0 innate immune activation. Now, one point that I wanted to highlight is also the chem path, that you actually see that there is kind of the only drug that across all 4 concentrations, you see some non-near 0 response. And the reason for that is not really because of the immunogenic profile. It's actually because it targets the CD52 or monocyte and dendritic cells, driving a target-mediated APC activation, leading to IL-6 release. So in this case, it's based on the drug mechanism that results in the cytokine release. So it's also important to understand the drug mechanism as well, to see whether is this something that is reasonable or expected for us to better understand what the result is. So then the big question for everyone to think about, don't really have a good answer for this yet, it depends, is when does your biotherapeutic or complex generic peptides become a clinical risk? That would be a good question to think about. So here we talk about, in this example, you can see actually there's a correlation between the in vitro and the clinical data here. And I know the earlier slide shows that aggregation is a factor where you can capture the innate immune response, but this particular assay captures beyond just the aggregation factor. So your post-translation modifications, your whole cell contamination, anything that is built inside your entire drug product, any potential innate immune triggers, including the process-related impurities, could actually present itself in this assay itself. So it's actually quite a comprehensive assay. It's not pinpointing a specific driving factor that leads to a cytokine release. It captures everything that can potentially lead to a cytokine release. So it's great in that sense. And here, it's also quite important to note that this is a comparative analysis. What I mean by that is that if you just test a one test article, in this case, and you see like maybe 100 picograms per ml for your IL-6 response, what does it mean? It means nothing, right? You need to have a comparator. You need to have something to benchmark against to make that result meaningful. So in this case, one example is that given chem path, is a high responder, Erbitox being a near zero response, your drug product can actually fall somewhere in between. And this can allow you to justify that your drug product, when this happens, is that it's within a controlled level of risk or relatively low immunogenic risk. So that can be a justification that you can use. But of course, depending on the drug that you are working on, you can consider different approaches. One is that whether are there a market-approved competitor, which you can then use as a benchmark. And of course, that's where you test whether your drug product is your equivalent or lower risk as compared to a comparator. Or another way is that if there's no suitable comparator, you can look within your pipeline itself. Maybe you have multiple candidates, and you can, based on that, to identify a lead candidate, which is of the lowest immunogenic risk. So it really depends on what stage of development you are and what exactly is the question you're trying to answer. Last but not least, this assay is the closest you can get to the clinical setting as you test with the undiluted threshold blood. So now moving on into what does the regulatory things, I think Dr. Daniela has actually covered quite a bit of this. It's basically back in 2014, there's actually guidance for the immunogenicity assessment for therapeutic protein products. So I've given the highlights here as mentioned here. So human whole blood is recommended in vitro assays to assess cellular activation and cytokine release potential. And negative animal findings do not override the positive in vitro signals, which shows that it actually goes back to the second slide that I have, whereby animal data doesn't translate very well into human data. And if we see something that is in the in vitro assay using undiluted threshold blood, that should, of course, take precedence over what you see in the animal data. So human relevant assays are essential in this case, and cytokine panel should include IL-2, 6, 10, interferon gamma, and TNF-alpha, which is already in our cytokine panel that we test. And moving a little bit along the timeline, going to 2023, there is a guidance that talks about the non-clinical evaluation of the immunotoxic potential of the pharmaceuticals. So here, it basically covers a range of drug products such as your small molecules, your oligonucleotides, and biologics as well. And here it mentions to recommend cytokine release assay using soluble and immobilized formats with whole blood, which we provide both formats as well. And I think this is really the key here. Assay selection should be fit for purpose based on the molecule being evaluated. The principle of the assay remains unchanged, right? You just look at the cytokine readout, you just incubate test article into the blood itself. But the study design is actually where it's critical to see if it can address the question you're trying to do. This will actually be covered a little bit more in the specific example of how we design the study for generic peptides, which is here. So before I go on to the study design part, I think it's important to talk about a little bit about the backdrop of how the agency thinks about the immunogenicity assessment of generic peptides. It all started with the UNDA guidance in 2021, and then it was quite simple. You just test 3 batches of your drug product against 3 batches of the reference listed drugs. And if you show no higher response than the RLDs, then you're fine. So the study design remains straightforward, 3 versus 3. And when we did this in the early days, we see that there's near zero response. And then after a period of time, the agency starts to wonder whether this near zero response, is it a true near zero response? What if your assay is just not sensitive enough to pick up any of these innate immune triggers? Because if it's an insensitive assay, whatever you test is going to be 0 anyway. So then the question is, how can we then capture this? So that's where we start to think about what are the suitable IIRMIs that can be included in the assay to demonstrate that sensitivity. So after that was done, right, you show that the assay is able to demonstrate sensitivity, we repeat it, you test 3 of your drug products against 3 batches of RLD. We see near zero response, great, and the assay is sensitive. Now the next question is, the concentration that you use to test in this assay, could it be so high that you kill all the cells to the point that you see near zero response? And the near zero response is not because it reflects the immunogenic profile of your drug, but it's just because the concentration is so high, you kill every single cell in the assay itself, which means you will not have any response. So with that kind of evolutionary thinking, we have actually updated the assay along the way to capture the current FDA expectations. And this is our recommended study design for generic peptides. So here you have your pilot study, which is also a dose finding study, to identify what is the best concentration that we can proceed to the main study where you compare 3 batches to 3 batches, or 3 batches of drug product to 3 batches of RLD. Now, the best concentration in definition here, it is the highest concentration that does not impact your cell viability. So here is a pilot study where we recommend just a 10 donor study because this is just it's just a dose finding experiment. It's nothing serious. So we recommend a small study. And we just have to test one batch of your drug product just to see what would be the optimal concentration. And in this case, the concentration we are doing is based on the dilution factor from the stock concentration of your drug. So here we are talking about like 1:5 dilution. In this case, it's basically 1 part of your drug in 4 parts of blood, leading to 1:5. So 1:10 means 1 part of your drug to 9 parts of the blood, and so on and so forth. So we test across 4 dilution factors, and we also recommend to include the excipient itself as a test article, just to see if you do see any cytokine response. It will be good to understand if that's coming from the formulated drug product itself, or is it actually it also comes from actually excipient itself, the formulation buffer itself. It will be good to have that information when that happens. So after you have identified the concentration, the next thing is the main study. That's where you need a larger cohort of donors of 20 to 30, and then you evaluate 3 batches of drug product against the 3 batches of IIRMI. In this case, we integrate the APC viability as well at the determined concentration from the pilot study. And here, to demonstrate assay sensitivity, we include 5 IIRMIs here, which is LPS, FSL1, Clo75, Zymoxan, and MDP. So testing at a range of concentration at the presence and absence of drug product to demonstrate assay sensitivity. Now, the absence of DP makes sense, right? It's just to test whether the assay is able to capture these IIRMIs on its own. Now, the presence, which means spiking these IIRMIs into the drug product, is important because the agency wants to see what is the interaction between the IIRMIs and the drug product itself. Like, more importantly, does it have any suppressive effect? Because if there is any suppressive effect, you could potentially see low response as well, but not really because of the drug itself, but because of the IIRMI's ability to dampen that cytokine response. So this is a data of our 5 IIRMIs here. So you see that we actually test it at the range of concentration. So this is actually selected to kind of demonstrate a titratable concentration and to show that the response actually goes down across the concentration to demonstrate their sensitivity as well. And this will also be reflected in this table, which is easy for the agency to understand how sensitive this assay is. And just to add on, right, so there are 2 types of sensitivity that we talk about here. Assay sensitivity, where what is the lowest concentration of cytokine you can capture. In this case, ours is 100 picograms per mil, which is the lowest level of quantification. And there's another one, which is the IIRMI sensitivity, which is what is the lowest concentration of IIRMI in the assay that can provide a detectable cytokine response, which is at least 100 picograms per mil. So there are 2 types of sensitivity here, which is then also presented in the report, which is for the agency to review. So here I talk about the integrated APC viability analysis. So the importance of this is also highlighted before. The agency is concerned if your response is near 0, the question is whether or not is this really a reflection of the risk profile of your drug, or is it just because the cells have all died? So we actually include a number of antibodies to actually look for different markers such as CD45, CD3, CD20, and CD14, HIDR, and CD16. So we look at 4 different APC subsets. That's really based on a lot of trial and error before we understand what are the APCs of significance to the agency. So we are looking at B cells, monocytes, dendritic cells, and neutrophils. And these are the different markers that we look at, so we kind of get into it. So one example would be like your B cells, we look at CD45, and then afterwards we look into CD20 positive and CD3 negative, and then after that we look at the live date viability stain. In this case, we are looking at the low frequency because these stains are only uptaken by the dead cells, so you want it to be as low as possible to show the viable cells. And this is repeated for the different subsets as well. So in terms of the regulatory recognition, we are actually the first to submit the Type 5 drug master file to the US FDA for our cytokine release assay. So this helps you to simplify the reference process for your abbreviated new drug application, also ANDA for short. So it allows it, in the drug master file, it captures the detailed protocol, the SOP, and validation data. So it makes it easier for the agency to better understand the different aspects of the assay, which is not something that we can share directly due to confidential information that's inside. And then we have also been invited as a subject matter expert to the US FDA and CRCG, which is known for Center for Research on Complex Generics. So this is back in October 2024 that this happened. And we are now quite confident to say in the innate immunogenicity assessment side, we are familiar with, we are kind of up to date with the latest FDA expectations of innate immunogenicity. And in the recent year, there haven't been any new updates to it. So it's kind of more or less a done deal, I would say, until, of course, there's new discovery to it. So some of the success stories that we have is vasopressin, glucagon, and liraglutide. So the success stories in this case basically refers to sponsors that have actually used our data and managed to get successful approvals with agencies. But beyond these three molecules, we have also have experience with teriparatide, tirzepatide, teriparatide, tirzepatide, and some other more that at the moment I can't think of. But yeah, basically we have a lot of experience with generic peptides. So this one is actually more of a sales speech slide where we actually share about how our partners, why our partners choose to work with us. So we actually provide, rapid turnaround time. We are quite accessible. Me and Emma, we are just one WeChat away. And we are also industry leading and also innovative. In fact, we are the first to commercialize a lot of the assays that we have actually, both Emma and I have actually described today. And also for scientific guidance, given the amount of expert in this room, I can't really say it's really guidance. I would say it's more of like a discussion where we can actually explore ideas together. And then communications, yes, we can always discuss more. And I think the most important part is a single point contact. So like for both Emma and myself, we actually handle more than just the sales side and the technical side. We can also help with contractual side, shipping side of things. So a one-stop, one-man solution, if you will. And last but not least, we also do regular visits to the APAC site. So we are more than happy to actually visit you at your companies. And then we can always have in-depth discussions for specific projects that might need further discussion. And last but not least, this is a testimonial from Dr. Reddy's. They have been a great collaborator. It's awesome, and we hope to continue that way as well. So yeah, that's all of my slides. Thank you. Any questions? Okay, there is someone for a question.
Thank you very much for the talk. Actually, I just remember the residue discussion. I just remember from previous talk. Is it possible that, for example, I have a generic peptide drug. I know the molecule that I designed, but because for when we do CMC, we only detect the things we know and we can. We don't detect the residues we don't know for impurity check. With this test, can we actually kind of goes back to what type of impurities?
Right.
Unexpected ones in the final product.
I understand. That's really a good question. So this is something that I feel that for innate immunogenicity is slightly better, is that you don't have to identify the impurities. So earlier, a lot of the talk about impurities, we are talking about like the peptide-related impurities. So these are impurities that needs to be identified, characterized specifically, and then you then do the subsequent adaptive tests. For innate immunogenicity side, it's a holistic kind of assessment. You don't have to identify what are these driving factors. Yeah. So it depends on your perspective, right? The good thing is that if I test the entire drug product itself, anything that is a potential innate immune trigger will show up in this assay itself. So if you see a near zero response given all these things are addressed, then it's great news. But in the event you see a moderate response, then we have to think about what would be the potential driving factors. Is it the process-related impurities driving the response? Is it the excipient itself? Is it whole cell contamination? Is it the PTM itself? So that will become the question for later after you see the response. So it really depends on perspective. The good news is that you don't have to identify what the impurities are. At least for generic peptides case, there is no specification of what are these innate immune the IIRMIs basically. You don't have to identify what are these innate immune triggers. They just expect you to test holistically your 3 batches of your drug product against 3 batches of RODs. It's only the adaptive side, the peptide-related impurities, that the agency expects you to identify and characterize them.
Okay, thank you.
Can I ask another question? Then, if you see the signal, what do you do next? If you see the signal when you identify the signal, from this ProStorm reaction, what do you do next?
Sorry, I might not have catch the earlier part. Sorry, could you repeat?
When do you see when do you see the consequences of ProStorm reaction?
Okay.
So what do you do next then? Because if you don't know which peptides exactly caused this ProStorm.
Right. That's a good question. So for this particular part, it will really be no. I understand the question, but I don't really have a good answer to it because it really comes down to what kind of what level of understanding we have of the drug molecule itself, and we have to think about the drug mechanism itself to why this happened. Is it something that's expected or intended? Because sometimes when you see a cytokine response, it doesn't necessarily mean that it's immunogenic. It could be part of the mechanism. So I don't really have a good answer to that at the moment. It really depends on the drug itself.
So maybe I can try to answer that. I was just going to try and answer that a little bit. Can you hear me? Yeah. So two things. One is the reason why at least the FDA doesn't ask you to identify impurity is the thought that there could be multiple impurities working together. There can be synergism. So it's really difficult to try to identify. And we don't think it's useful. We think that, it's just as long as you can control the innate immune activation, the agency doesn't need to know where it comes from. What to do next? There's a couple of papers published on that. We published some, and some other people published that. It could be aggregates. It could be other things, but there are different tests that can be run, different cell lines that have specific receptors that can help you orient as to what kind of impurities are there. But as he said, that's sort of a second stage to help you then clean up the product and make sure. But the first thing is to understand whether you have a problem or not, which I think this assay that does quite well. It's just after that, then you need to figure out, do a root cause analysis, and that may require some additional testing. And it's going to depend a lot on what your product is, what platform is made in. Again, you have to understand the whole product, the whole process to really go fishing for what's wrong and what's going on.